previous lecture, Mendel came up with this whole concept of genetics, right? That we were passing traits to our offspring. He didn't really know what to call them yet. But he viewed them simply as either dominant or recessive. Either you blocked everything or you were hidden. Okay, so we then began to realize he was initially criticized, as I mentioned, for it being simply too simplistic. So he did, and to a certain extent, they were correct. And in fact, we call traits that are single gene traits with a true dominant, true recessive nature, Mendelian traits after Gregor Mendel. So we have variations of that. So incomplete dominance. This is when something where one allele is not completely dominant over the other. And the heterozygote actually is an intermediate phenotype between the two parents. An example of this, snapdragons, also Japanese 4 o'clock flowers are a classic example. You've got red, you've got white, and then the heterozygote is actually pink. Okay, and then when we go through here, right, and you'll notice over here that we're writing things a little bit differently than when we do our Punnett squares. You need to do this when you're doing cases of incomplete dominance, because it's different. It's not, notice how there are different letters, whereas when we do Punnett squares, it's one letter and one's capital and one's lowercase. Okay, and then the phenotypic ratio would be 1 red to 2 pink to 1 white. Codominance. Both alleles contribute to the phenotype of the heterozygote. Roan cattle, you either have red cattle, you have white cattle, or you have this guy here, which is roan, which is a combination of both red and white. Okay, you can have this in chickens, right? We've got this dude that's black, white, and then we've got this sort of checkered rooster. Okay, and then if we were to go through here and cross a chicken and a rooster together, right? Then you see this BB, BW, BW, WW. Okay. Multiple alleles. Some genes have more than 2 alternate forms. Okay. ABO blood type is the classic one that we go with in humans. Okay. And what's really interesting here is A and B are codominant to one another. Okay. Which is why you can get an AB blood type. And then O is recessive to both A and B. Okay. So then when we look here, and again, notice how I have this capital I, capital A, and then this lowercase I actually indicates O because it's the recessive. Whereas this IB, then, those guys are codominant. And that's where it can get a little bit tricky in the Punnett squares. Okay. So the immune system produces blood proteins that can cause clotting when a different type enters the body. Okay, you kill people. Okay, until we knew how to blood type, it was sort of a last resort. Or some people, like, it was this big, like, you felt rejuvenated, but then some people died. So that's bad. Okay. Oh, this shouldn't be the feathers. Sorry. Anyway, but we're going again here. Okay, if I do an A with an AB, right? O isn't a possibility. We'll do this in class in person. Pleiotropy, a single gene has a whole bunch of phenotypic effects. Okay, sickle cell anemia is an example of this. So we think of it as your red blood cells are sickle shaped, they can't carry oxygen as well. But the problem is you're not just physically weak, you don't just typically have anemia with low oxygen levels. They can also clump, which can cause brain damage, it can cause pain. They can accumulate in the spleen. They can potentially cause damage to other organs. So there's a lot going on. Sickle cell anemia isn't just something where people have a hard time breathing. It actually is a great deal more complicated than that. And then we have polygenic traits, which are traits that are controlled by many genes. Okay, so we have we think we have about 3 genes that control skin color. Okay, and this is where anytime you get a polygenic trait, you're going to get this bell-shaped curve, okay, associated with tones. And if you ever walk down the makeup aisle at a store, you can see the wide variety of tones that there can potentially be. This didn't used to be. I know that for many, many years, it was a very hard time for women who were much darker complected of African and Asian descent to be able to find makeup because it just simply wasn't marketed towards them. I'm very lucky. I just go and pick out whatever the palest color that there is, and then I go with them. I'm going to show you this video in class. This is a really interesting one. Okay, this is an interracial couple, and they have 2 sets of twins that are one is very definitively Caucasian and the other is very definitively African. Eye color is also a polygenic trait. Okay, we always like to do the blue eyes versus brown eyes things. That's because blue eyes is recessive to everything else. Blue eyes is actually the lack of pigment in your eyes. Okay, but this is why you get this wide range of everything from dark brown to hazel to gray to green to amber. I've seen people with eyes that they still are sort of classified as brown, but they're a lot more yellow in them, to blue. Now, based on Mendel's observation and genetic studies, it was determined that chromosomes were actually what were transferring this genetic information. Prior to this, we thought it was proteins, because proteins do every other single stinking job in the cell, so why not this one too? So genes are located at specific positions on chromosomes, right? And then the behavior during meiosis and fertilization account for the inheritance patterns. We have our homologous chromosomes that separate during meiosis. I've got independent assortment. I've got crossing over. And then fertilization results in the restoration of the diploid number of chromosomes. And actually, that is where it came from. Walter Sutton was studying grasshoppers, and he noticed that grasshoppers' body cells had 24 chromosomes in them, and their gametes only had 12. And he's like, oh, maybe, and he was really the first to derive it. And then we'll go into detail as to exactly, when we talk about DNA replication, we'll talk about how we actually figured out that it was chromosomes that was carrying this information. This is just a nice little diagram talking about the law of segregation versus the law of independent assortment. I'd like it. It's just a nice visual for you guys. So certain genes are linked. If they're linked, they tend to be inherited together because they're really close to one another, which means crossing over doesn't happen frequently between them. So for example, if you have a white cat with blue eyes, they're deaf. Okay, that tends to be carried together. And in fact, if you have a white cat that has two different colors eyes and one of them is blue, they will be deaf in that ear. There are some in humans. I have a cousin whose little girl, her ureter, which is the tube that comes from the kidney down to the bladder, was not in a great position. And so it caused basically reflux into the kidney, and it was causing kidney damage. So they had to do surgery and improve it. And it seems to be most frequent in redheaded females. So again, it seems to be a linked trait. So we've talked about crossing over that can produce different combinations of alleles. Okay. And then Thomas Hunt Morgan did all this with Drosophila melanogaster. Okay, fruit flies are great. They stink to high heaven, but they're great. Okay, they have a 2-week reproduction time, which is awesome. They only have 4 chromosomes, which makes them really easy to study. Okay, they breed like rabbits, and they're very easily cultured. Like you literally just put them in test tubes with food at the bottom, and they just grow and grow and grow and grow, which you know if you've ever had a problem with fruit flies. Okay, so he looked at crossing over and inheritance, and there are lots of really weird variations of fruit flies. Okay, so 2 linked genes can give rise to 4 different gamete types. Okay, so we've got all of this going on together over here. And then he used this in order to actually locate genes on a chromosome using linkage mapping. Okay, and what they did is they looked at, Sturtevant was a student of Morgan's. The further 2 genes are apart, the less likely they are to be linked, the more likely crossing over is to happen. Okay, so if we look here, right, we've got this short antennae versus long. I've got gray versus black body, red eyes versus cinnabar eyes. You actually can see the color difference. Normal wings, vestigial wings, these guys can't fly. Okay, and then I've got red eyes versus brown eyes. So they can be used to map positions on the chromosomes. Okay, and this is actually how we initially did a lot of our genetic mapping, was looking at crossing over. And you guys are going to have to do this. So recombination frequencies. So if I look at the percent of recombinant offspring, then I can tell how far apart those are. And really nicely, 1% equals 1 map unit. They just were like, let's make this simple. So if I go here, Okay, I say the recombination between cinnabar eyes and what is this body color is 9%. Cinnabar eyes and vestigial wings is 9.5. Between body color and vestigial wings is 17. Well, this is the only order that they can potentially be in. Okay. Like I said, he called them MAP units. It's referred to as a centimorgan after Morgan's lab. It's 1% recombination frequency. It made it really nice, easy. When you make up stuff, you get to make the conversion easy. Okay, so if I look here, I've got black body, short wings are 36% recombination frequency. Between purple eyes and short wings is 41%, and between black body and purple eyes is 6%. So what I would traditionally do, okay, I would take my biggest one here. I'm going to draw that first, okay, and then between here and the body is 6%, and here is 36%. It's not always going to add up to this. And the reason is we're talking percentages. Okay, so there are rounding issues, plus or minus 2%, and you're okay. Okay, so like I said, also crossing over, like if you have two crossing over events, it can, multiple crossing over events cancel them out. Okay, so that's going to reduce the number of recombinant offspring, so that's going to alter our percentages too. Okay, basically, if you have greater than 50% recombination frequency, they act as if they're found on separate chromosomes, and they are considered to be inherited independently. Now, sex chromosomes and sex-linked genes. So this can have a big impact here. In mammals, females are XX, males are XY, right? Males become male because of turning on the Y chromosome. Okay, the genes get turned on. Default in humans is female. Okay, all human embryos start as female, and you can have individuals who are genetically XY but develop physiologically as female because the Y chromosome never turns on. Okay, the absence of Y chromosome allows for ovaries to develop. So that's the big deal here. We've got other things in other animals. So for example, my cute little grasshopper here, XX is female, just X is male. In chickens, the heterozygote at the sex chromosome is actually female and the homozygote is male. And in bees, if you're diploid, you're female, and if you're haploid, you're male. Okay, so any gene that is located on the sex chromosome is considered a sex-linked gene. So here's the deal. There's not a whole heck of a lot of crossing over between X and Y is significantly smaller than X and lacks numerous genes that X carries. So basically, if you're a female, you need to have 2 copies of the allele to have this particular deficiency. If you're male, you only need 1 copy. Okay, so was discovered during work with fruit flies. So we looked here and we noticed, okay, so we've got red eyes, right? And then if I cross a female who is heterozygote, all of a sudden with a male who has red eyes, all of a sudden this white eye showed up. Okay, and it's because the Y chromosome doesn't carry anything to block. That's why we see here the R that is written sort of as an exponent. Notice how it is not next to the Y. It's because the Y doesn't carry that gene. Okay, so a male receiving the X-linked allele from his mother will have the disorder. A female needs to receive it from both parents. So hemophilia is our classic example of a sex-linked trait. Hemophilia is a problem with clotting factor 9. People tend not to clot. They tend to bleed out. And so this was referred to as the European royal disease. It was rampant in European royal families because they were all sort of related to each other because you couldn't marry outside of your station. You married another royal family. So basically you're marrying cousins. The youngest of the Tsar's children, Alexei, had hemophilia, and they sort of elsed it. They sort of locked themselves up because they were fearful for his life. They knew it could potentially kill him, and they actually think that was part of what caused the downfall of the Russian Tsars. Rasputin was able to feed off a lot of the fear in the community, and that really led to it being overthrown. The one other thing I'm going to talk about is color blindness. Red ring color blindness is sex-linked. Okay, so Greg is colorblind. That doesn't mean he can't see any color. It's just he sees colors differently. And so it must have come from my maternal grandmother's side, but I don't know much beyond that because my dad is not colorblind. So he couldn't have passed the trait to me. It can't come from your father. Okay. So it wouldn't matter whether my husband was colorblind or not. It doesn't come from him. My mother's father is not colorblind. So it didn't come from him. So it must have come from my mother's mother. And she's an only child. To her knowledge, her father was not colorblind. So it's probably further back. So you could get these that are hidden for a long time. And that completes the patterns of inheritance.